Once you have an understanding of what the organelles of the cell are and what they do, it's important to be able to use that knowledge and apply it to a model in a lab. So this is one of the lab models that we have, and we should be able to take the information we've learned when we drew the cell and identified its parts to transfer that knowledge and apply it to this model. So let's start with the most prominent part of the cell. We know that that would be the nucleus. So this circular organelle here is the nucleus. You can tell it's the nucleus because it has the nuclear pores, these little orange circles. But it also is cut open, which shows you on the inside the nucleolus that is responsible for producing ribosomes. And it shows the DNA, but it doesn't look like X's because all of that DNA is sort of cut open. So you're just seeing all of these light blue areas as the DNA. Now on the outside of the nucleus, we have these membranes that have little blue dots all over it. And those blue dots are showing you the ribosomes. So if there are ribosomes on these linear structures outside of the cell, then we know that that is the rough ER. So now we want to look for the smooth ER. We know the smooth ER looks similar to the rough ER. The big difference is there are no dots or ribosomes on its surface, so it looks smooth. So all of these components over here, these are all parts of the smooth ER because they look like the rough ER except no ribosomes. So they would be called smooth ER. Next, we have the Golgi apparatus. We know this is the Golgi apparatus because it has these kind of flattened stacks together and then also has these vesicles surrounding it. Remember that the Golgi vesicles are like bubbles that can transport things around the cell because the job of the Golgi apparatus was to package things. We have our mitochondria over here. We know that mitochondria looks kind of oval shaped and it has this squiggly line in it. So remember when we drew that out, what it looked like. So now we know that that's what it is on this model. We have our plasma membrane or cell membrane, which is just the barrier that keeps certain things in and certain things out. On this model, it shows you some cilia or hair-like projections that would be on the outside of some cell surfaces. We also have our centrioles. Remember, centrioles are made of microtubules, so that's why they look like lines, and they are important for cellular division. They always are going to occur in pairs. Now, probably the one that's the most challenging to apply your knowledge to is these sort of bubbles, because we know that these can be lysosomes or peroxisomes. Lysosomes would take care of worn cell out parts, while peroxisomes would neutralize toxins that could be in the cell. We are going to label these as lysosomes on this model. Now, what you can't see but is here is the cytoplasm. Remember that cytoplasm is just the fluid of the cell that all of the organelles are suspended in. So we could just say it's there on its surface. So now that you have seen how to draw the cell and you have applied that to the model, go ahead and go practice. Could you label these parts on your own?